we are about to begin our special lecture this afternoon. I'm very honored to introduce Professor Bertrand from France. He has been a friend, and we worked together on some topic on the theme of autism before. But he has been a long-term friend and serving as an advisory board member for our former NRPGM national projects, and currently is the NRPB advisory board members. And Professor Jordan has a long history of research and achievement. He worked for the Marseilles Genome Pool, and also currently now on the advisory board of the CORE Bio PACA facility. So today he will talk about the topic is autism, the elusive gene. So let's welcome Professor.
Well, thank you very much, Daniel, for the nice introduction. Yes, I think we already, or at least with some of the audience, we already discussed this topic maybe 3 years ago, but it has grown and changed quite a lot, so I thought it was worthwhile to go back. I'm not a specialist in psychiatry, definitely not. The reason I got interested in autism is that first there was a story in which I became involved, which you will hear about. And also I think it's a good example of the complexities of diseases that have a strong genetic component, but yet where we have a lot of trouble finding the genes involved because there are many genes and the effect of each of them is quite small. And it's also a good example of the way in which the biotech industry and basic science and the public interact, not always in the best possible way. I'm also quite involved now. I mean, I'm basically a molecular biologist and a genomic person, but I've also been involved quite a lot as an advisor to biotech companies. So this is a world which I know, I'm beginning to know very well. So, and actually this is the title of my talk is the title of a book that will come out in France in January, which will be my 11th book about science, genetics, and so on for the general public. I hope it will eventually be translated into several languages, including Chinese. And actually, by the way, I'm very proud to have had one of my books translated into Chinese. This is it. It's called the Love Song of the Sea Cucumber, and it's been published in mainland China, but I hope it will eventually be published in Taiwan as well. And it's really a book about the evolution, or around the topics of evolution. Okay, so autism, the elusive gene, actually is better than the untraceable gene. Well, that's how the story started for me, with an article in France, in Le Monde, which is I would say, our most respected newspaper, a bit like the Times in England. So that was an article 5 or 6 years ago, announcing that a French company was going to launch a diagnostic test for autism. That's my translation, of course. And so it announced that this company was going to launch a genetic test for autism. These are the actual words of the company on the company's website. And the journal, the newspaper also said this would be a home test, which sounded quite strange. I mean, a home test like a pregnancy test. And actually this is what you could find at that time on the website of the company, and indeed they did announce the world's first genetic test for autism, allowing the identification of individuals at a very early age and to be launched very soon. This was summer 2005. It was supposed to be launched early next year in the US. Okay. So at first sight, this looked quite surprising and a little disturbing, quite surprising, because from what I knew at the time, autism was very complex, and no major gene had been found, and it seemed very unlikely that one could produce workable, reasonable genetic tests. And also disturbing because, of course, in such diseases, one has to be very careful about what is announced. People, the general public tends to believe that genes determine everything, and you can reinforce that belief by such announcement. And also, home tests. Sorry, I have to go back. No, I'm going forward. And I mean, home tests seemed like something very unlikely. Well, let me clear that up immediately. This business of home tests was a mistake on the part of the journalist. Presumably, he had been talking with company officials, and they had said something about home brew. Home brew means a test that is done in the company's own lab, not sold outside as a kit. So this is called colloquial term is home brew. The official term is LDT for laboratory developed tests. And so apparently the journalist had mistaken home brew for actually a home test, something which you do at home in your kitchen. So this is actually wrong. But this made the news even more strange. Okay, so this is what was announced on the website of the company. So let's go briefly over autism, because maybe not everybody is familiar with it. Just some basic facts. So the picture I showed, and which is shown here again, is actually from the Wikipedia piece on autism, and it shows a presumably autistic child who has one of these manias. In his case, it's putting all his toys in a very strict arrangement before he can go to sleep, and presumably if one of the toys is out of line, there's no way he can go to sleep. This is one of the symptoms, but there are many others, and basically these people have trouble having social relationship, acquiring language, and so there are, of course, difficulties being inserted into society, and they very often are not able to lead an independent life. And the prevalence is quite high, at least using a fairly wide definition. There's a lot of discussion. The prevalence has grown a lot in recent years. There's some discussion about whether this is really increasing prevalence or just increasing attention and wider diagnostic criteria. But anyway, it's in the range of 1 and 150, which is pretty high. Okay. Now, particularly in France, this is a very sensitive topic because, as you may know, in France, we still have a very strong influence of psychoanalysis as a, well, it's supposed to be a science. I don't think it's a science, but that's a matter of debate. And anyway, autism is, by many people, including many MDs, still considered as an infantile psychosis, and according to, well, to a certain extent, to the inventor of the term, Leo Kanner, who published the first paper in 1943, but also to Bruno Bertrand, who is well known here, Françoise Dolto, who is, well, better known in France, I guess, than over here. Autism is supposed to be the result of difficulties in the relationship between the mother and the child. The mother somehow doesn't recognize the child as a person or as a child, and the child retreats into isolation, into this, what Bertrand called the empty fortress. So the prevalent view of autism, at least in France, is still that it's a disorder of the mother-child relationship. maybe it's no longer the prevalent view among psychiatrists, but it's still very widespread in the general public and in nurses and people like that. And it has, of course, pretty serious consequences for the children involved and also for their parents who are charged with a responsibility which is not really theirs. By the way, we have lots of time, so if at any time you feel a question, please ask the question right away, because by the end of the talk you might have forgotten it. So by now we do know that there is a very strong component in autism. This has been shown most convincingly. Sorry, I still have. Still clumsy with this. This has been shown most convincingly by looking at the concordance in real twins and false twins, in monozygotic twins and dizygotic twins. So concordance means you look at a twin pair. One of the twins has been diagnosed as autistic, and what you look for is whether or not the other twin has also been diagnosed as autistic. And what is found is that between monozygotic twins, the concordance is very high, and between dizygotic twins, the concordance is significant, but is not larger than for any siblings in the same family. So this is a very strong indication for the strong influence of genetics, because whether the kids are monozygotic or dizygotic twins, they are born together from the same mother, they are raised in the same environment. So presumably the environment is as similar for two dizygotic twins as it is for two monozygotic twins. And so if the concordance is much higher for monozygotic twins, it must mean that the genes are involved, because of course monozygotic twins have almost exactly the same gene alleles, not quite, but almost, while dizygotic twins only have on average 50% of shared alleles. So if there is such a strong genetic influence, then of course one expects that one should be able to find genes for which some alleles protect and some alleles predispose to the syndrome. This is just some of the original data. One of the first papers, it's just part of a figure of a paper published in 1985. So you have here several sets of siblings. The monozygotic twins are shown by this linkage between the two symbols. And so this is a monozygotic twin pair, this is a dizygotic twin pair, and of course the affected individuals are shown in black and the unaffected ones in white. And as you can see, almost all dizygotic twin pairs are concordant, which means if one of them is affected, the other one as well, while most dizygotic twins are non-concordant. Sorry. This is only part of the data. There is more data in that paper. Actually, there have been one or two papers very recently which lower a little this concordance, but still the concordance is much higher for monozygotic twins than for dizygotic twins. And this is an indication that there is a genetic component, either genetic vulnerability or really genetic quote-unquote determination. So monozygotic twins around 85%, dizygotic twins around 10%, siblings also around 10%, and of course general population 1%. Apart from genetics, there is also some physiology. it's now possible to do functional brain imaging to find out which regions of the brain are activated during certain tasks, like showing a child animals and asking him to name them, or tasks like that. And it seems now really established that you have significant differences between the areas that are activated for a certain task in normal individuals and individuals that have been diagnosed as autistic. And well, generally speaking, this is very complicated data, and I'm also not a neurobiologist, but generally speaking, this seems to be related to problems of connectivity between synapses, between the neurons. And it's visible very early. So again, this confirms that autism is definitely not a psychological reaction to some kind of trauma. It's really a physiological problem, a neurophysiological problem with some gene alleles giving a strong probability. Okay, in terms of therapy, as I'm sure you know, there's no really highly effective therapy for autism. There are many phony therapies around. This is one which again has quite a strong following in France and has had a lot of negative exposure. Recently, something called packing, which involves wrapping the autistic child in cloth, in sheets that have been kept in the refrigerator, and then letting the temperature go back to normal while he is being talked to by therapists. This is supposed to be, by its proponents, it is said to be effective in cases of automutilation, which is one of the complications of autism. It has no scientific basis whatsoever. You have things like this saying that vaccination is the cause of autism. You can see this picture where the hand inoculating those poor children is this is highly spectacular. You have also quack doctors like this that claim to cure autism by various things, like this Dr. Chun Wong. You have more serious therapies, behavioral therapies, two major methods, one called TEACH, one called ADA, which through fairly intensive behavior therapy can indeed succeed in making those children more capable of interacting and of following some kind of school curriculum and have better integration. But there is no miracle. It's definitely not extremely effective, but it can help. And those are actually the only method that have undergone reasonable scientific evaluation and that have shown some effectiveness. Okay, so much for, the general presentation of autism. Well, the only thing on which everybody agrees is that whatever therapy one is using, it should be done very early as possible. Yet, presently, the diagnosis for autism is really reached by the age of 3 or 4, which is very late, which is the time by which brain plasticity has more or less disappeared. So indeed, if a good genetic test was available, it would probably be useful because it would allow to start therapy very early and to get better results. So there is a real, besides scientific reasons, there is a real reason for trying to find genes involved in autism or in vulnerability to autism. So what has been done? So now I'd just like to paint a picture as it was by the time this announcement was made, which is 2005. I'll come back later to what has happened since in terms of scientific work and discoveries. So, as you may remember, psychiatric genetics has been very much in fashion already in the 1990s, and generally it's been a miserable failure. After the first successes of medical genetics, finding the genes for muscular dystrophy and cystic fibrosis and Huntington's disease, then there was a lot of excitement and a lot of work trying to find the genes involved in, sorry, I have trouble finding the names, for example, schizophrenia, in manic depressive syndrome, and all those psychiatric diseases for which there is indeed a genetic component, and autism was among the lot. And I would say, as with the other psychiatric diseases, these efforts failed in that they found many loci, different loci in different studies, sometimes conflicting results when the same set of patients was analyzed by two different labs, and generally fairly low statistical significance. Even at that time, it was clear that only blood scores above 3 were likely to be somewhat significant. You have to remember that by that time, these are all genetic mapping exercises, looking at the number of cases, controls, and trying to find alleles that are, well, markers that are associated with the disease. And at that time, this game was played with a pretty small set of markers, something like 200 markers to cover the genome, which was enough to find the gene for Huntington's, or the gene for the gene variant for Huntington's, or the one for cystic fibrosis, but definitely not enough for something complex like autism. That was the 1990s. Well, by the 2000s, we were able to use so-called microsatellite markers. If you remember those, we had a pretty good genetic map with a few thousand markers all over the genome. So genetic mapping got a bit better, but the results were still quite tentative, contradictory, and the situation was still very unclear. So indeed, this is a review published in 2006, so it more or less summarizes the state of research by 2005. That is by the time this announcement was made by this French company. So it did seem very strange that a company should be able to announce a genetic test for autism, given those results. Okay, so this actually summarizes what I was saying. So now let's look at the announcement of the company. So this is in 2005. This is also from the website at the same date. So basically what they say is that their test looks at 4 genes. So it is an attempt to take into account the fact that the genetic component of the syndrome is complex. And well, basically what they say here is that in the most extreme case, where one child has all the risk versions, the risk alleles, then his or her relative risk will be around 50, which means this child would have a risk which is 15 times the standard risk in the population. So this sounds fairly impressive. But actually, if you look at the figure, it was announced also, for example, on the sites of autism charities, like Cure Autism Now, which I think has changed name now, but anyway, autism gene isolation, using samples from that charity. So this is the actual paper. The announcement was based or was simultaneous, if you want, with a scientific paper published by the company's scientists. These are all people working in that company, IntegraGen. And actually this paper was published in quite a good journal. This is a journal with an impact factor of 10, so it's quite respectable. But as you can see, both the title and here the conclusion of the paper are quite careful. Haplotypes in the gene PRKCB1 are associated with autism, and the conclusion, our data provide evidence that the PRKCB1 gene may be involved in the etiology of autism. So this is a careful scientific conclusion, supported by the data in the paper, but it's of course quite far from being an autism gene that's going to be able to predict. So to do this, they studied a reasonable set of families. They have a special mapping method developed in that company. They found 7 chromosome regions that appear to be associated with autism, and the most significant of them contains this gene, which looks like a good candidate because it's expressed in the brain and it's involved in synaptic transmission. Well, unfortunately, they didn't see an alteration of this gene in patients, which is usually what really clinches the results in such studies. So, as I said, their conclusion is careful. But as we saw, the announcement from the company is much less careful. And indeed, if the relative risk is 15, what they say, my slide arrangement is not very good, but anyway, what the company says, what was in the previous slide, is that if all 4 genes have the quote-unquote bad allele, then the relative risk is 15-fold. And actually the test, the use they propose for this test is first in families where there is already one autistic child, to test the siblings to find out if one of them is at high risk. But they say also that they hope that eventually the test will be improved and can be used to screen any child who looks slightly abnormal. And again, they announced that the test will be marketed in 2006, which means 6 months, basically, from the date of the announcement. And indeed, if what they say is correct, well, positive test in such a case would mean a 15-fold relative risk of 15-fold higher risk of autism. If we take the standard risk as 1 in 150, as we saw before, that means a positive child, according to the test, would have a risk of 15 times 1 over 150, which means a risk of 1 in 10. Which means that actually 9 out of 10 so-called positive children would actually be false positive. So this doesn't look like a very good test, because of course you want a genetic test to give you few false positives and also few false negatives. And in this case, if you label